A frontier is the extreme limit of understanding or achievement in a particular area. Frontiers are at the edge of our understanding, and yet there is something so calling about them. People have always wanted to push our understanding further and further and create new solutions for existing problems. This is the trait that allowed humans to survive and thrive, from discovering land to pushing social frontiers, and eventually pushing the boundaries of science, giving us more insight into how our world works. This was the exact mindset in which the secret of life was discovered, the structure of DNA. It took 80 years for people from around the world to help solve one of the secrets of life, deoxyribonucleic acid, also known as DNA. It revolutionized life as we know it, creating an entirely new frontier of science revolving around genetics and heredity. It also made numerous breakthroughs in medicine, genetics, and even nanostructure. It all started with a monk named Gregor Mendel, who, in 1865, did an experiment with peas outside of his monastery. He realized that when breeding peas, certain traits were passed on while others weren't. He coined these terms recessive and dominant traits, a term that was used to identify major genes and their functions. 4 years later, in 1869, Johann Miescher stumbled across an unusual chemical while trying to study white blood cells and pus in order to research more about tuberculosis. Thinking nothing of it, he named it nuclein because of its position in the nucleus of the cell. This molecule would be later known as DNA. The rise of the 20th century produced physicists like Albert Einstein and Niels Bohr, who dazzled the modern audience with their scientific advancements in chemistry and other sciences. Biology, however, was taking a backseat in the worldview of scientists. Miescher and Mendel's research was overlooked and almost forgotten. However, one of the debates in the small community of scientists was about DNA, whether it or proteins were responsible for gene replication and traits between generations, something that no one on Earth knew how. If anyone was able to find the answer, it would create a new frontier. The majority of the scientific community at the time theorized that there were proteins in the structure of the cells. There were so many proteins in humans that it was thought to store genetic data. This was fueled by the increase of interest in the frontier of eugenics, a field about the study of genetics and how to purify genes. However, Oswald Avery's experiment in 1930, where he studied the effects of DNA in genetics by isolating bacteria strains and checking how they passed on the DNA from each strain, and how the DNA controlled the strain's traits. This revolutionary experiment proved that DNA could contain genetic information. This experiment changed the mind of two people that would be crucial in order to figure out the structure of DNA. There were 5 scientists who were primarily focused on finding the structure: Rosalind Franklin, James Watson, Francis Crick, Linus Pauling, and Maurice Wilkins. They all started to work on this problem at the same time and were split into groups: Watson and Crick at Cambridge, Maurice and Rosalind at King's College, and Linus Pauling, 5,000 miles away at Caltech. In 1951, Linus Pauling was the world's most renowned chemist and had become a world master at solving complex molecular structures. In his lab at Caltech University, he was able to oversee the chemistry division and make groundbreaking research. He thought that genes were located in proteins in cells and theorized on this problem. At King's College, Maurice Wilkins was working with Rosalind Franklin, and he wanted to produce images of DNA using X-ray crystallography. Maurice Wilkins was a close friend of Crick's, and they corresponded with each other on the research regularly. At Cambridge, Watson needed a partner, someone who was also an expert in X-ray crystallography and could help him with the research. That person was Francis Crick. They hit it off instantly and agreed to find a method of attack to solve the puzzle. First, the 2 main parts of DNA are the sugars, adenine, thymine, guanine, and cysteine, and the phosphates, deoxyribose. Arranging the sugars in a certain way stumped scientists, and even if that problem was solved, there was still the issue of the phosphates, whether it or the proteins were on the inside of the structure. In 1952, 2 separate events happened that were crucial in order to discover the structure of DNA. Rosalind Franklin took a picture of DNA called Photo 51. This revolutionary photo changed the game, as when Crick found the photos, he recognized the shape and determined that DNA was in fact a helical structure. The next event happened later in November when Pauling shared his theory to the world. He apparently had solved the puzzle of the structure of DNA, showing that it was in a triple helix form. This crushed the spirits of Watson and Crick. Their only redeeming factor was that if they were able to find evidence independently which supported Pauling's theory, they might get some credit in the paper that was going to be published. In early February of 1953, Pauling's paper was published, and when Watson and Crick read it, they thought it was now officially over and they had lost this race. However, they found that Pauling's model was a rush job. There was not enough clear-cut evidence to support the theory that the structure of DNA was in fact a triple helix. This gave them new hope, and they realized that they could build their own models, and that the race was not yet over. And finally, on February 28, 1953, Watson had an epiphany. He cut out the shape of the molecules on cardboard and tried to arrange them in a way that they could be bonded together. Then he finally saw it. The bond between adenine and thymine, and guanine and cysteine. Over the next few days, he and Crick finished their model, the double helical structure of DNA that we all know today. This model helped to solve all the problems of heredity. You've inherited one half of your DNA from your mother and the other half from your father. The sugars that made up inside DNA provided instructions that could be read by cells in your body to create proteins that determine every physical attribute in your body. Watson and Crick's paper was released 14 days later, on March the 14th of 1953, in the Nature magazine. For the first few months of it being published, it gained a lukewarm response, but over the years, as more people decided to pursue projects in genetics, this discovery was crucial in order to help rebuild an entirely new science, with the structure of DNA as its foundational idea. During the 1970s and 80s, it helped to produce new and more powerful scientific techniques, especially recombinant DNA research, genetic engineering, rapid gene sequencing, and monoclonal antibodies, techniques on which today's multi-billion-dollar biotechnology industry is founded. It also founded the principles of modern molecular biology, which studies the composition of cellular molecules that carry out biological functions in the human body. After the discovery, researchers could examine the molecular mechanisms and structures responsible for such complex processes. With all this new information, new drugs could be created to target bacterial infections, now that we know how they work on a molecular scale. The research came in the early years after the discovery was made, so Watson and Crick themselves were involved in many of the new projects that were coming in, earning Watson and Crick Nobel Prizes in 1962. In 2003, the Human Genome Project was completed. Humans finally sequenced all of the genes in the human body, opening the door for more advanced gene therapy, creating new cures for diseases such as cancer, and for the first time, the ability to manipulate genes and therefore their subsequent traits. It laid the groundwork for CRISPR, where you can edit genes by precisely cutting DNA and then letting natural DNA repair processes take over. It is in itself an entirely new frontier that reduces the effect of genetic diseases and defects. DNA profiling, DNA fingerprinting are all based on correlating organic material to a person based on their DNA. From a material/structural point of view, double-stranded DNA has a 2 nanometer diameter and each turn is 3.4 nanometers long. It is an ideal building block for making biocompatible, biostable, and nanometer-scale constructs through molecular self-assembly in 2-dimensional or 3-dimensional spaces. Designer DNA nanostructure can serve as a molecular pegboard to arrange different external elements, like proteins, peptides, metallic nanoparticles, DNA aptamers, and small organic compounds. All of these processes came to fruition in 2021 during the coronavirus pandemic, when an RNA vaccine was used to counter COVID. Foreign RNA from coronavirus interacted with ribosomes in human cells. This method could also help protect against variants of COVID. RNA-based vaccines are able to stop mutations to the virus too, because it creates a layer of herd immunity through a population. In 1865, Gregor Mendel did not know that he was going to discover an entirely new science, something that forever changed how our world works. Multiple events led to the culmination, the revolutionary discovery of the double helix. Our sheer determination to expand past the limits of what we know and into the unknown makes the thrill of discovery so much more worth it.